Hi students, we have to meet once again to discuss a new lesson in botany, not in zoology, botany, genetics. Chapter number 3, Chromosomal basis of inheritance. In the last session you see that one, we have studied something about the classical genetics. In that one we have studied about the Mendelian inheritance, etc. In this lesson, we have to discuss something about deviation of concepts related to Mendelian principles. Any deviation from the Mendelian principles we have to discuss. For example, Mendel did not explain what is called the crossing over, the phenomenon of linkage. These are all deviations from what is called the Mendel's principles. So we have to study deviation of concepts related to Mendel's principles. And also the chromosomal theory of inheritance. Now Mendel, you see that one studied the defined characters of garden pea plant and proposed the law of heredity. But his work was not disclosed because of various reasons, not recognized. But in 1900, 3 different people revealed the Mendelian concepts. You know the people, Hugo de Vries, Carl Correns and von Scherman. The later periods, because of advancement in microscopy, now cytologists studied the internal organization of nucleus. They found some worm-like structures present inside the nucleus. They are called as chromosomes. The name is given to the chromosome because colored bodies. Once a stain is given, a dye is applied, it exhibits color. Chromo, color. Soma, body. So colored bodies, they will take color after staining procedure. If we are taking each chromosome, it contains long, coiled, thread-like DNA molecule, in which are arranged, you know that one, the genes in a linear order. The genes are the hereditary units. They have a definite position on chromosome, what is called the locus, what is studied about the characteristics in zoology. Okay, and they are considered as hereditary units, have specific what is called actually locus, arranged, having genes arranged in a linear way. Now what is the meaning of chromosomal basis of inheritance or chromosomal theory of inheritance? The chromosomal theory of inheritance normally states that the Mendelian factors, what we can say the genes, have definite locations, definite position. Such positions are called the locus. If we are taking a chromosome, we have the genes arranged in a linear way. The point where a particular gene is located is called as a locus. That is called, for example, first locus, second locus, third locus. Gene A is present the first locus, gene B is present the second locus, like that. So locus is a point in a chromosome where a particular gene is located. So now this theory states that now we have the Mendelian factors. These factors have definite locations called the locus. And the genes, they carry the information from one generation to another generation, from one generation to the next generation. That is a law. So the chromosomal theory of inheritance states that the Mendelian factors or the genes have definite locus, that is a position on chromosomes, and they carry information from one generation to the next generation. This is what is called the law of or chromosomal basis of inheritance. Now we have to go for the historical aspect of chromosomal basis of inheritance, or the cytological findings, the important cytological findings related to, that is chromosomal basis of inheritance. A few people. Okay, I'll take that one. Now let's see the historical development of chromosomal theory of inheritance. So the following actually important cytological findings related to chromosomal theory of inheritance have been developed. Number 1, a person by name Wilhelm Raux in 1883 He suggested that the chromosomes in a cell are responsible for transferring heredity. That is information. Number 2, Montgomery in 1901. He was the first to suggest that the occurrence of distinct paired chromosomes, the chromosomes are found in pairs, that was suggested by Montgomery. He also suggested that the maternal chromosomes form a pair with the paternal chromosomes only during meiosis, only during solution. Otherwise, that's created here and there. So the pairing of maternal and paternal chromosomes occur only during meiosis, that was suggested by Montgomery. Then the famous two people, one by name D. Theodore Boveri in 1901, suggested the idea that the chromosomes carry or contain genetic determinants, otherwise called, you know that one, the genes are the factors. He was largely responsible for the development of chromosomal theory of inheritance. Number 1, he supported the idea that the chromosomes contain genetic determinants or the genes or the factors. He was largely responsible for the development of, that is the theory of chromosome theory of inheritance, along with another person by name Sutton. We'll see about that person, after which we have to go to the lesson. Another person who is related to what we call the chromosomal basis of inheritance or chromosomal theory of inheritance, that is W.S. Sutton. In 1902, he recognized a parallelism, that is nothing but the similarity between the behavior of chromosomes and the Mendel's what's called the factors, the Mendelian factors. He just identified what is called the similarity, otherwise called parallelism. That is nothing but the similarity between the behavior of chromosomes and the Mendelian factors during gamete formation. So both together act as one unit. There are certain similarities between these two, that is between the chromosomes and the factors, what we can say gene. So he recognized a parallelism or similarity between the behavior of chromosomes and Mendelian factors, nothing but the genes, when during meiosis or during gamete formation. Now Sutton and Boveri independently developed or proposed the chromosomal basis of inheritance. These were the two people responsible for the development of what we have the famous chromosomal basis of inheritance in 1903. Okay, they were responsible, others contributed, but these were the people largely responsible for the development of chromosomal basis of inheritance. Now Sutton united the knowledge of chromosomal segregation during meiosis and the Mendel's principles and called it what we have the chromosomal theory of inheritance. He united the knowledge of what's called chromosomal segregation or separation that occurs during meiosis and the Mendelian factors or the Mendelian principles, what we are following, the one gene, one factor, what is called theory, monogenic inheritance we can say. So Sutton united the knowledge of chromosomal segregation during meiosis with what we call the Mendel's principles and together call it as the chromosomal basis of inheritance or chromosomal theory of inheritance. So if the question arises who are responsible for the development of a chromosomal basis of inheritance, we can say Sutton and Boveri. This theory is also called a Sutton and Boveri theory of chromosomal basis of inheritance. Now we have to go for the various characteristics of the salient features of the theory, chromosomal basis of inheritance. What are the salient features of chromosomal theory of inheritance? You know we have the somatic cells. The somatic cells are derived from the zygote by the repeated mitotic divisions. These somatic cells contain two sets of actually chromosomes, two different sets of chromosomes, or we can say two identical sets of chromosomes. Two different in that sense, that is different. So two identical sets of chromosomes. One set of chromosome is received from the female parent, another set is received from the male parent. The one which is received from the female parent is called the maternal chromosomes. The one which is received from what is called actually the male parent is called paternal chromosome. These two chromosomes are called as homologous chromosomes. If you are taking a pair of chromosomes, one chromosome is received from the female parent called the maternal chromosome. Another chromosome is received from father, that is a paternal chromosome. These two chromosomes are similar in size, length, shape, arrangement of genes, etc. Hence called homologous chromosomes. So the homologous chromosomes are exactly identical in length, size, shape, the arrangement of genes also. Okay, and such chromosomes are called, that is homologous chromosome. There is the first character. One set from female parent, another set from the male parent. These two chromosomes are called homologous chromosomes in a pair. Now, the chromosomes actually retain their original structure, their uniqueness, individuality throughout the life of an organism, throughout the entire life of an organism. No change occurs in that one except a mutation. Then, each chromosome has specific determinants. What we can say the Mendelian factors, now we call that one as a genes. The third one, specific determinants, are present in each chromosome. They are nothing but the Mendelian factors. They are called factors by Mendel. Now it's called as the gene. Then the behavior of the chromosomes during what's called the gamete formation just to provide an evidence to the fact that the genes are carried on the chromosomes. We are getting the proof or evidence only through what we have the gametic formation. So during meiosis, behavior of the chromosome and the behavior of the genes normally more or less similar. That is called parallelism. So the behavior of chromosomes during gamete formation that provide an evidence to the fact that the genes are carried only by the chromosomes. These are all the salient features of chromosomal theory of inheritance. Now we have to go for the experimental proof in support of what is called chromosomal basis of inheritance. Though the chromosomal basis of inheritance was developed by Sutton and Boveri, it was confirmed by T.H. Morgan, Thomas Hunt Morgan from America. In 1910, he was the man just actually gave a proof for chromosomal basis of inheritance. So, chromosomal basis of inheritance was developed by Sutton and Boveri and proved by T.H. Morgan in Drosophila. Let's see the evidence. So regarding the chromosomal theory of inheritance, discussions have gone and also controversies by different scientists all around the world. But the debate has been cleared experimentally on an animal by Thomas Hunt Morgan. So Thomas Hunt Morgan in 1910 confirmed what we call the chromosomal theory of inheritance. Confirmed the chromosome theory of inheritance by taking the animal Drosophila melanogaster. That is what's called the fruit fly in 1910. Now the Drosophila completed its life cycle in 2 weeks. It has 4 pairs of chromosomes or 8 chromosomes. Now he has taken what is called actually the character related to the eye color. In Drosophila, several shades of eye color, more than 8 shades of eye color. But the red eye color is dominant over other eye colors. Now he took Drosophila eye color. 2 varieties he took to explain the chromosomal theory of inheritance. Number 1, red eye color. Another one, white eye color. The genes for these characters, eye color, are located in X chromosomes. So it is an X-linked character and follow the sex during what is called inheritance. So in Drosophila, 4 pairs of chromosomes. Gene for red or white eye color is located in X chromosome. And so also we have, for example, yellow body color and miniature wing. These two are also located on X chromosome. But if you are taking actually, you have studied sex-linked characters. The males are hemizygous. So now it is comparable to the hemophilia. It is an X-linked character, as in the case of hemophilia or in the case of color blindness. Now normally it follows crisscross pattern of inheritance. Suppose if the male is white eye color, then we have in F1 generation we have red eye color. Suppose cross occurs, I will represent cross occurs between a carrier female and white eyed male. We will get, as in the case of color blindness, 50-50 male and the no just actually white eye color appearance in Drosophila. Similarly, I can say it follows a crisscross pattern of inheritance as in the case of color blindness and hemophilia. I will represent in the next slide. So, in the case of color blindness also in human beings, we have actually the males are hemizygous, having only one gene. Having only one gene in the X chromosome. Suppose the X chromosome, this is the Y chromosome. If you have the gene A, there is no counterpart. There is no corresponding allele in the Y chromosome. That's why given the gene has no counterpart in Y chromosome. If you have the genes A, there is no corresponding allele in the Y chromosome. That's why the males are called hemizygous. The same statement what I mentioned in color blindness. Whereas the female, they have two X chromosomes. They have, for example, two alleles. XX, two alleles. Either both are dominant or one dominant, one recessive. So anyway, normally the females have two alleles for that character. The males have only one allele for that characters. Recall color blindness, the same mechanism here also. Instead of color blindness, we are taking what is called the eye color. Red eye color is dominant, corresponding to a normal person. White eye color is recessive, corresponding to color blindness. So like that we have to compare that one, the same mechanism. I will explain later now with the illustration. So there are two alleles in female while the male has only one allele. Here I represent. This is the one. Okay. So the genetic results completely based on what we have the behavior, the meiotic behavior of X and Y chromosomes. Which one carries the gene to the next generation? Whether the genes are present on X or Y chromosomes, accordingly we can just explain. You see that one, the genes are carried by the chromosomes. So it is an experimental probe only to show that the genes are carried on the chromosomes and we have the genes and chromosomes behave similarly during the gamete formation. So the genetic results completely based on the meiotic behavior of X and Y chromosomes, what they behave during meiosis. I will show that illustration. Like the eye color, red and white eye color, we also have, for example, the genes for body color, yellow body color, and miniature being. Miniature being. Miniature means small. So the gene for yellow body color and miniature being also present the X chromosome. These support the chromosomal theory of inheritance. They support actually strongly the genes are carried only on the chromosomes. Now the genes, the linked genes connected to the sex chromosomes is called sex linkage. The linked genes connected to the sex chromosomes is called sex linkage. For example, this is called sex linkage. The genes connected to the sex chromosome either to X or to Y is called what is known as sex linkage. Okay, so yellow body color, miniature being, also X-linked character in support of what is called chromosomal theory of inheritance. What is chromosomal theory? I mentioned already. The genes have definite locus in the chromosomes. They carry information from one generation to another generation. So during gamete formation, the behavior of the gene and the behavior of the chromosome will be the same. That is, they exhibit parallelism and similarity. Now I will show the illustration, that is an explain with the diagram. T.H. Morgan proved the chromosomal theory of inheritance by taking the character, the sex-linked character, namely the eye color in Drosophila. There are many shades of eye color. He took red eye color and then white eye color. Now red eye color is normally dominant over white eye color and it follows what is called the sex during the inheritance. It is comparable to the color blindness. Let's take the red-eyed female normal. then white-eyed male, that is colorblind. In that way you have to compare. Normally what is happening, the red eye is dominant. In the case of female there are two X chromosomes, so we have two alleles in female. But in the case of Y, we have only one X chromosome and just we have to represent only one allele, either dominant allele or recessive allele. When a cross is effected between red-eyed female and white-eyed male, these are all the parents. In the F1 generation, all the individuals are red-eyed, irrespective of the sex. That means both the males and females are red-eyed. You compare this one. Suppose father is colorblind. You know, just the sons are normal. Here the red eye is normal. I am saying that one comparison only. If father is normal, the son would be colorblind. Here, just you see that when the male is white eye, then definitely just in the F1 generation, all the males are what is called red eye color, opposite. I mentioned already there is no compromise between father and the son. If father is white eye, definitely the son would be red eye. Compare color blindness. Now, here all the females are carriers. You see that one capital R, one small R genes, dominant and recessive genes. Now this is the F1 generation, all are red, both males and females. So male, father, the first one white-eyed male. When father is white-eyed, then definitely the son would be red-eyed, as in the case of color blindness. Father normal, son would be color. Now let's take what is called a carrier mother. Now here the red-eyed female, this is a carrier. It produces the eggs. 50% of the eggs with capital R and 50% of the eggs with small r. Now similarly, if we are taking just one male, red-eyed male. So we are making a cross between red-eyed female carrier and that red-eyed male. So 50% of the males have what is called the dominant gene and 50% of the males have no genes, that is what we call the Y chromosomes. So 50% X and 50% Y chromosomes, Y chromosome no genes, only that is the females have R because the X is only having the gene because the males are hemizygous. Now we are making a cross between a carrier female, a carrier red-eyed female and what is called a normal red-eyed male, So once mother is carrier, as in the case of colorblind, once the mother is carrier and father is normal, you know that all the females are normal. And among the sons, 50% normal, 50% colorblind. Recall that one. The same manner, here the female is carrier, red-eyed. The male is also red-eyed. That is comparable to normal. So, carrier and then normal. As a result, in the F2 generation, we have received 4 individuals All the females are red-eyed because mother is carrier and father is normal. I am using the word comparison only. Here no father, no mother. So I am saying that one. So all are red-eyed. That's why the females are all red-eyed. As per the male's concern, as in the case of color blindness, because here the female is carrier and just actually you see that when the male is red-eyed, 50% of the males are red-eyed and 50% white-eyed. So 50-50 only here. You compare this one, that is what with the color blindness. Here also there is what we call crisscross inheritance. So red-eyed female, then carrier female transmitted to red-eyed males. Okay, so normally this is happening, crisscross inheritance following the just what is called the inheritance as in the case of color blindness. So here the genetic results actually based on the meiotic behavior of X and Y chromosome. They get segregated along with the genes. They unite at random to form what is called a specific sex with the specific characters. So this indicates that actually it's a strong proof showing that the genes are carried on the chromosomes. Because in that one Y chromosome has no genes. You can say simply the genes are carried by the chromosome. Another example what I mentioned we have the yellow body color and miniature. They are also responsible for what we call sex-linked characters. So these two characters strongly support what we call the chromosomes carry the genes or the genes are located on the chromosomes. Now here, the linked genes are connected to the sex chromosome. These are the linked genes. The genes which are linked. The linked genes are connected to the sex chromosomes. And this is called sex linkage. The linked genes are connected to the sex chromosome. That is called sex linkage. Now here I mentioned already in the case of Drosophila, there are 4 pairs of chromosomes. Now out of these 4 pairs, we have 3 pairs are called somatic chromosomes. Somatic. The remaining 1 pair is called the sex chromosomes. 3 pairs of somatic chromosomes, 1 pair of sex chromosomes. In the case of male, the X chromosome is long and straight, whereas Y chromosome is short and bent at one end. If you are comparing the female Drosophila, the somatic chromosomes are more or less similar, both in male and female. But in the case of female, both the X chromosomes are long and straight because Y chromosome is absent. The difference between X and Y, X is long and straight, Y chromosome is short and bent. So there are 4 pairs of chromosomes representing what we call the sex linkage, nothing but the genes linked to or connected to the chromosome, and this is called sex linkage. So the linked genes connected to the sex chromosomes is called sex linkage. Now go through the diagram. Okay, so it is following the same principle as in the case of just what we call color blindness. Remember, compare the red-eyed female is normal, so red eye is normal and white eye is what is called color blind. So if white eye here, then definitely the sons. This is father, the sons, we have white eye, then opposite character. Then a cross between a carrier and what is called a normal male. Definitely, if the male is normal, all the females are actually normal. That's here referring to the red eye. Among the males, 50% are red eye and 50% are white. So I am telling once again, compare this one with that of what is called color blindness. You can get the idea easily. Okay. So it follows crisscross pattern of inheritance. Okay, thank you. I will get to the remaining part of chromosomal basis of inheritance in the next class.